Hello everyone. The topic we are going to discuss is glycogenesis. The word itself tells you a lot. Glyco means sugar or glucose, and genesis means creation or formation. So glycogenesis literally means the creation of glycogen. Let us understand what that means, why it happens, and how it happens step by step. I am Brindaronika, tutor working at Triple M College of Nursing. Before we get into the topic, let me tell you what we are going to cover. First, we'll define glycogenesis, then we'll understand why it is important, the significance of this process. After that, we will identify the site, meaning where exactly in the body does this happen. Then we'll go through all the steps of glycogenesis one by one with the enzymes involved, and finally we will look at the energy involved and the overall significance. Okay, let us begin. Think about this. Every time we eat food, especially carbohydrates like rice or bread, our blood glucose levels goes up. The body absorbs all that glucose, but the body cannot use all of it immediately. So what does it do with the extra glucose? It stores it, right? And the way it stores it is by converting all those extra glucose molecules into a large branched storage molecule called glycogen. This process of making glycogen from glucose is called glycogenesis. Now, why is this important? See, our body needs a constant, steady supply of glucose, especially the brain and the red blood cells. They are completely dependent on glucose. So the body is very important. When there is excess glucose after a meal, it's stored it as glycogen. And when blood glucose falls later during fasting or between meals, that glycogen can be broken down to release glucose back into the blood. So glycogenesis is essentially the body's way of saving glucose for later. It's like a charging a battery when there is extra power. So you can use it when needed. Now we'll see the definition of glycogenesis. Glycogenesis is defined as the process of glycogen synthesis in which glucose molecules are added to chains of glycogen for storage. So, glycogen is a large, highly branched polysaccharide, meaning it is made up of many glucose units all linked together. When the body has excess glucose, it keeps adding glucose units one by one to build up this large glycogen molecule. The entire process of building up glycogen from glucose is what we call glycogenesis. Now let us talk about why glycogenesis is so important. Actually, there are four main reasons are there. The first reason is that it maintains blood glucose concentration during fasting, starvation, and when we eat limited carbohydrates. This is especially important for the nervous system and erythrocytes, that is the red blood cells. The brain and RBCs depend entirely on glucose, so having a glycogen reserve is critical for their survival. The second reason is that glycogenesis helps maintain adequate concentrations of intermediates of the citric acid cycle. This keeps the overall metabolism running smoothly. And the third reason is that it helps clear the products of metabolism from other tissues from the blood. For example, lactate is continuously produced in muscle cells and RBCs during exercise. Glycerol is continuously produced in adipose tissue when fats are broken down. All of this can be converted and used for glycogen storage, which clears them from circulation. And the fourth reason, and this is a clinical point worth remembering, excessive activity of this pathway can occur in critically ill patients who are responding to injury or infection. In those situations, glycogenesis, which feeds into glycogen synthesis, is highly active, and this can contribute to hyperglycemia in those patients. So as nurses, understanding this helps us monitor blood sugar in ICU and critically ill patient. Now, where does glycogenesis take place? Glycogenesis takes place in the cytoplasm of cells, specifically in the muscles, liver and adipose tissue. The liver is the main site. Liver glycogen is used to maintain blood glucose levels for the entire body. When blood glucose drops, liver glycogen is broken down and glucose is released into the bloodstream. Muscle glycogen, on the other hand, is a local energy reserve. It is used only by muscle cells during exercise and physical activity. Muscle glycogen cannot directly release glucose into the blood. It is kept for the muscle itself. And adipose tissue also has some glycogen, though in smaller amounts compared to the liver and muscle. Alright, now we are going to go through the most important part, the steps of glycogenesis. There are 5 main steps are there. Let us go through them one by one. Step 1 is glucose uptake. Before any glycogen can be made, glucose from the bloodstream first needs to enter the cells. So the first thing that happens is glucose is transported from the blood into the liver or muscle cells. Now this transport does not happen randomly. It happens through specific carrier protein called glucose transportase. In the livers, the transportase used is GLUT2. In muscle cells, the transporter used is GLUT4. This is a process called facilitated diffusion. Glucose moves from higher concentration in the blood to lower concentration inside the cell. Through these transporter proteins, this ensures an adequate supply of glucose inside the cell of glycogen synthesis. Now, as soon as glucose enters the cell, it immediately gets phosphorylated, meaning a phosphate group is added to it. This is done by the enzyme hexokinase in the muscle cells and glucokinase in the liver cells. The glucose becomes glucose 6-phosphate and ATP is used in this reaction. So, what is happening here means glucose plus ATP gives us glucose 6-phosphate plus ADP, and the enzyme is hexokinase in the muscle or glucokinase in the liver. This traps glucose inside the cells so it cannot go back out. Step 2 is a conversion to glucose 6-phosphate. Now we already started this in step 1. Once glucose is inside the cell, the enzyme hexokinase or glucokinase add a phosphate group from ATP to the 6th carbon of glucose, forming glucose 6-phosphate. Why is this step important? Means because by adding a phosphate group, the glucose becomes negatively charged and can no longer pass through the cell membrane. It is now trapped inside the cell and committed to being processed further. This phosphorylated form, glucose 6-phosphate, is also a key branch point in metabolism. It can go into glycolysis to make energy, or it can continue the pathway toward glycogen synthesis. So to say it simply, glucose plus ATP gives glucose 6-phosphate plus ADP, and the enzyme is hexokinase or glucokinase. Now step 3 is the isomerization to glucose 1-phosphate. Now glucose 6-phosphate has the phosphate group on its 6th carbon, but for glycogen synthesis we need it on the 1st carbon. So the phosphate group needs to be moved. This is done by the enzyme called phosphoglucomutase, which transfers the phosphate group from the 6th carbon position to the 1st carbon position, converting glucose 6-phosphate into glucose 1-phosphate. So this is called an isomerization reaction, which means the molecular formula stays exactly the same, but the position of the phosphate group changes. And why does this matter? Because glucose 1 phosphate is the correct form needed for the very next step actively by UDP. UDP means uridine diphosphate. So what is happening in this step means glucose 6 phosphate becomes glucose 1 phosphate with an enzyme called phosphoglucomutase. Step 4 is the activation of glucose, where glucose 1 phosphate is converted into UDP glucose. Now here is something important to understand. Before glucose can be added to the growing glycogen chain, it needs to be activated, meaning it needs to be attached to a carrier molecule called UDP, which stands for uridine diphosphate. The enzyme that does this is UDP glucose pyrophosphorylase. It transfers the UTP molecule uridine triphosphate to glucose 1 phosphate. This produces UDP glucose and inorganic pyrophosphate. Now, UDP glucose is the activated, energized form of glucose. Think of it like glucose that has been loaded onto a delivery truck. Now it's ready to be handed over and added to the glycogen chain. The release of pyrophosphate makes this reaction irreversible, which means the process is strongly committed to moving forward toward glycogen synthesis. So, to say it simply, glucose 1 phosphate plus UTP gives UDP glucose plus PPI, and the enzyme is UDP glucose pyrophosphorylase. Step 5. It has two parts. The first part is glycogen elongation. Now we have UDP glucose ready. The enzyme glycogen synthase, which is a key regulatory enzyme of the entire glycogenesis pathway, now takes this UDP glucose and adds it to the existing glycogen chain. How does it add it? It forms a bond called an alpha 1,4 glycosidic bond between the new glucose unit and the end of the growing glycogen chain. Each time this happens, so the chain gets one glucose unit longer. So the chain keeps growing one glucose at a time, with each new glucose unit being donated by UDP glucose, and each time a glucose is added, UDP is released as a byproduct. This continues until the chain is long enough for branching to occur. So mainly, to say it simply, UDP glucose plus glycogen with residues gives glycogen within plus 1 residues plus UDP, and the enzyme is glycogen synthase. The second part of step 5 is the branching of glycogen. Now, if the glycogen chain just kept growing in one straight line, it would be very long and difficult for enzymes to access all the glucose units quickly. So the body creates branches to make glycogen more compact and more accessible. When the glycogen chain reaches a certain length, about 11 glucose unit, the branching enzyme steps in. The branching enzyme is called alpha 1,6 glucosyltransferase. It cuts a segment of about 6 to 7 glucose units from the end of the chain and transfers it to an internal position on the same or a nearby chain. It attaches the segments using an alpha 1,6 glycosidic bond instead of an alpha 1,4 glycosidic bond. This creates a branch point. Now both the original chain and the new branch can continue to grow by glycogen synthase. More branching happens as the molecule grows, giving glycogens its characteristic tree-like highly branched structure. Why is branching important means because the more branches glycogen has, the more free ends there are. And glycogen synthase can only add glucose to the free ends. So more branches means faster synthesis and also faster breakdown when needed. So this is about the second part of step 5. So now look at this diagram. It shows the complete pathway of glycogenesis. Starting from glucose at the top, hexokinase or glucokinase in the muscle and liver converts it to glucose 6 phosphate. Then phosphoglucomutase converts it to glucose 1 phosphate. Then UDP glucose pyrophosphatase convert it to UDP glucose. Then glycogen synthase adds glucose units to form alpha 1,4 glycosyl units, the long chains, and finally the branching enzyme creates the alpha 1,6 branches, giving us the final glycogen molecule with both alpha 1,4 and alpha 1,6 glycosyl units. On the left side of the diagram, you can see the actual structural changes. Glucose being phosphorylated, then glucose 1 phosphate, then UDP glucose, then the growing glycogen chain with branching enzyme creating the highly branched glycogen structure. Practice diagram and labeling this complete pathway. It's frequently asked question in exam. So let us now bring everything together. Glycogenesis is a tightly controlled process that enables the body to store extra glucose as glycogenesis. So it is just not a simple storage reaction. This process transforms glucose into a compact, highly branched energy stored that can be rapidly mobilized when the body needs it. Every step from glucose uptake to phosphorylation to isomerization to activation as UDP glucose to chain elongation and branching, each step has a specific enzyme and specific purpose. The regulation of glycogenesis is controlled by two key hormones. One is insulin, another one is glucagon. Insulin, which is released after a meal when blood glucose is high, promotes glycogenesis. It tells the body to store glucose as glycogen. Glucagon, on the other hand, is released during fasting when the blood glucose is low, and it inhibits glycogenesis and promotes glycogen breakdown instant. As nursing students, understanding glycogenesis helps us understand why patients with diabetes have problems with glucose storage, why glycogen storage disease occur, and why blood glucose management is critical in clinical setting. Thank you.